So, yes. So in the last class we have discussed what is invasion and what is metastasis and by what route the spread of tumors takes place. Right now, in this class, what we are going to study is what is the mechanism or what was the pattern behind this spread of cancer, spread of tumor cells. That is nothing but mechanism or biology of or patterns of spread of invasion and metastasis. That means here in this class we are studying the mechanism behind invasion and metastasis. Okay. So in this process it involves mainly different 8 steps approximately. So it is explained in the molecular level. So it includes the first step is aggressive clonal proliferation and angiogenesis. Aggressive means rapid or very fast clonal proliferation. That means proliferation or multiplication of cells. And angiogenesis. Angio means blood vessel. Genesis means formation. Angiogenesis means formation of new tiny blood vessels. Okay, so this was the very first step in the biology of invasion and metastasis. So what is clonal proliferation? Usually it includes, the first step includes spread of cancer cells that takes place very rapidly, and this is explained on the basis of tumor heterogeneity. What is tumor heterogeneity? That is nothing but, so in the population there is a multiple n number of tumor cells are there. Out of that, only a few population of clone of, like only a few group of these tumor cells, they have the capacity, they have the ability to undergo rapid proliferation. So these cells which has that capability, they undergo rapid proliferation, and this helps in the clonal proliferation. That means proliferation of n number of cells, n number of tumor cells. That cannot be done by all the tumor cells. Only a particular subpopulation or only a particular clonal cells have that capability to undergo rapid proliferation. So that is aggressive clonal proliferation. Clonal means a group of cells which has that ability to undergo rapid proliferation will be considered as a single clone, and that we call it as rapid or aggressive clonal proliferation. Okay, and then angiogenesis. So tumor angiogenesis is a very much important step in the process of metastasis. Why? Because in this angiogenesis, tumor angiogenesis, new tiny blood vessels were produced or were formed. So these tiny blood vessels which were formed, they are like they are in direct contact with the tumor cells, right? So they has the more susceptibility to undergo metastasis, and see, they are more vulnerable to invasion as these evolving vessels are directly in contact with cancer cells. So whatever the new blood vessels that were formed, they are in direct contact with the tumor cells, right? So as they are directly in contact with these tumor cells, they are more vulnerable to get invaded by the tumor cells. So there is more chance of these tumor cells to enter into these newly formed blood vessels. So that is angiogenesis. So first step is aggressive clonal proliferation and angiogenesis. And then tumor cell loosening. Generally in normal cells, one cell will be attached or sticked to another cell to form a tissue, right? So that attachment is done with the help of certain molecules called cell adhesion molecules. That is KCAM. Adhesion means sticking. So for one cell to stick to another cell, there is a glue-like thing that is nothing but cell adhesion molecules are there. But in case of tumor cells, there is deficiency of these cell adhesion molecules. Example of cell adhesion molecule is E-cadherin. So because of this deficiency of these cell adhesion molecules like E-cadherin and also of certain immunoglobulin superfamily, so because of this deficiency, what will happen? There will not be proper sticking or attachment between two different cells. So because of that, there will be loosening of tumor cells. So that results in loosening of cancer cells. And then third step is tumor cell and ECM interaction. What is ECM? ECM is nothing but extracellular matrix. So these tumor cells gets interacted with extracellular matrix. Okay. So whatever the loosened cells which were formed loose because of deficiency of this cadherin and any adhesion molecules. So they will become very loose. So whatever these loose tumor cells are there, now these will come and attach to ECM proteins. That is nothing but extracellular matrix proteins. Here mainly these ECM proteins include laminin and fibronectin. So when this tumor cell gets attached to this laminin or fibronectin, so because of that, there will be proper formation of a bonding between these two. So there is also loss of integrins and the transmembrane receptors for further favoring invasion. So when this stick gets bind to this laminin and fibronectin, during that time, whatever the integrins, transmembrane receptors, everything will be lost. So when these were lost, it will further promotes the invasion. So tumor cells, that is loosened tumor cells, they will bind with this extracellular matrix proteins like laminin and fibronectin. When these two together forms a compound or a complex, during that time there will be loss of integrins. And also transmembrane receptors were lost of the normal cells or the extra or these proteins. So which will further progress, which will further helps in the invasion. And then degradation of extracellular matrix. As we said that when these on these protein molecules like laminin and fibronectin, there is loss of integrins and transmembrane receptors, right? And now these tumor cells which are there, which were formed, that binds to these ECM proteins. So when this complex is formed, these tumor cells, what will they do? This will overexpress their proteases, matrix degrading enzymes, metalloproteases, collagenases, gelatinase. So all this cause the degradation of this extracellular matrix. So by forming a complex with this extracellular matrix, tumor cells, they will release certain enzymes which will destroy this extracellular matrix. And also the inhibitors of metalloproteinases were decreased. So another protease like cathepsin D is also increased in certain cancers. So these enzymes which were released, they cause dissolution or degradation of this extracellular matrix. So firstly, what will happen? Firstly, it will dissolute the basement membrane of the tumor itself, then make way for tumor cells to the interstitial matrix, and finally dissolve the basement membrane of the vessel wall. That means first, by releasing these enzymes like proteases, gelatinases, collagenases, what does it do? It will first initially, it will dissolute or it will cause the degradation of this basement membrane of the tumor cell is damaged. First, this basement membrane of the tumor cell is degraded, and because of this degradation, what will happen? It will make an access to enter into the interstitial matrix. So once it enters into the interstitial matrix, then it will cause dissolution of this basement membrane of the blood vessel wall. As newly formed blood vessels, angiogenesis. So whatever the newly formed blood vessels wall is there, that basement membrane will be dissoluted. So first, it causes degradation of tumor cell basement membrane, and then it gains access into interstitial membrane. So once it enters into interstitial membrane or interstitial matrix, so now it will cause a degradation of the basement membrane of vessel wall. Okay. So that is about degradation of extracellular matrix. It will be done with the help of certain enzymes like proteases, collagenases, and gelatinase and cathepsin D. And then entry of tumor cells into capillary lumen. So as I said in the last step, it causes degradation of basement membrane of blood vessels. So because of this degradation, it gains access into the capillary lumen. That is, in the vessel, it will enter into lumen of the blood vessel. So the tumor cells, after degrading, they are ready to migrate into capillaries or venules by following two mechanisms. First is autocrine motility factor. So autocrine motility factor, it's a cytokine derived protein. So name itself indicates autocrine motility factor. That means it will help in the motility of these tumor cells. That means it will help in the movement of tumor cells into the capillary lumen. And then cleavage products of matrix components which are formed from the degradation of ECM have properties of tumor cell chemotaxis. So in the degradation of ECM, so whatever the broken down products or the cleavage products are there, they will be having a certain properties of chemotaxis. That means movement of cells from one tissue to other. And also they have growth promotion of tumor cells, and they also promotes angiogenesis in the cancer. So these degraded products of ECM will help in the further growth of these tumor cells, their further movement, growth and angiogenesis, that will be done with the help of these cleavage products of extracellular matrix. So once all these tumor cells, once they degraded this extracellular matrix, entered into the basement membrane and then to the capillary lumen. So then they will enter into the capillaries or lumen of the lymphatic and capillary channels. Not only entering into this angiogenesis newly formed blood vessels, they also gain access into lymphatic vessels and capillary channels. So, and then thrombus formation. So the tumor cells which are present in this lumen of these capillaries, they are now covered with, so whatever they will be covered with circulating blood. So tumor cells that will be coated with the circulating blood in the blood vessel, and that will together forms a thrombus. Thrombus is nothing but clot. Tumor cells coated with circulating blood forms thrombus. So this thrombus, whatever it is formed, it provides nourishment to the tumor cells. So as we know that all the minerals, nutrients that will be supplied in the blood vessels through blood, right? So this whatever the blood supply that is having to these tumor cells, so that will provide nutrition or nourishment to these tumor cells, and also they will protect them from immune attack by circulating host cells. So whatever immune attack also will it will be protected from immune attack by the host cells by forming a coat over that tumor cells. So once these tumor cells, they are coated with circulating blood, the circulating blood will provide nourishment to these tumor cells, and not only that, as it is formed, this will not be visible to this our host immune system. So this will our host immune system is incapable of damaging or destroying this thrombus or tumor cells. Okay, and in fact generally only 0.1% of the malignant cells which enter into the bloodstream will survive. Remaining will be damaged or remaining will be destroyed by host immune system. Only 0.1% will be entered into this metastasis. Okay, this is about thrombus formation. So how is this thrombus formed? Tumor cells coated with circulating blood cells will form a thrombus. So these circulating blood cells, what will they do? They will provide nourishment to these tumor cells, and also they will hide or protect these tumor cells from the attack by immune cells or immune cells of the host. Okay, so that is thrombus formation. And then extravasation of tumor cells. So extravasation is nothing but once it is entered into the new blood vessel, now it will gain access into other vessels like capillaries, venules, and lymphatics. So this is how it will spread to different parts of the body. And then survival and growth of metastatic deposit. That is nothing but whatever the extravasated malignant cells are there, they get enlarged in the right environment. So when they get entered into a particular cavity or in a particular body part, so once it identifies, once it gets into a right environment, it will start further replication under the influence of growth factors. So growth will be done with the help of growth factors like PDGF, FGF, TGF beta, VEGF, that is vascular endothelial growth factors. So these were the growth factors which will promote further growth of these malignant cells or tumor cells. And not only these growth factors, but also cleavage products of ECM that we have discussed in the last step. So they also promote growth of these tumor cells. So these metastatic deposits grow further if the host immune defense mechanism is low. If your immunity is less, then it will multiply rapidly. And when our immunity also fails to eliminate that, then their growth will be very rapid. So these may further metastasis to the same organ or to other sites by forming emboli. So by forming emboli is nothing but when this thrombus gets dislodged from that region and enters into other part, then it will spread to distant places. And if it was found to, if it doesn't undergo like separation, or if it doesn't undergo formation of emboli, only the surrounding tissue of an organ will be get affected. So this is all about the patterns of spread. So I'm showing this picture now. So if you find this was the primary tumor. This is a newly formed blood vessel, that is angiogenesis. Newly formation of blood vessel was done, and this is this is the extracellular matrix. This is the lumen of actual blood vessel. So these were the tumor cells which were loosened because of the absence of cadherin molecules or adhesion molecules. These loosened tumor cells, once they get attached to these basement membrane of this extracellular matrix, what does they do? They will get interacted with this extracellular matrix. So thereby they gains access into this extracellular matrix. So once they entered into that, they cause degradation of this extracellular matrix by releasing certain enzymes like proteases, gelatinases, and all these enzymes. By this, they will cause degradation of this extracellular matrix. And then, so once there is damage of this extracellular matrix, they will gain access into the intracellular matrix, that is, they will enter into lumen of blood vessel. So all these tumor cells will damage this basement membrane. First, they will damage the basement membrane of tumor cells, and then damage the basement membrane of this extracellular matrix and gains access into this lumen. So once it is entered into the blood vessel, whatever the tumor cells, that will be coated with circulating blood in the blood vessel and forms a thrombus. Okay, so this is thrombus formation. So this blood red dots, that is nothing but blood. So this circulating blood will provide nourishment to this tumor cells, and also they will protect them from the protect from this defensive action of immune cells. And then, so once these were in this lumen, if it is there in that region, then it causes local spread or to that particular tissue, and then it enters into the circulation, extravasation of tumor cells. That means they will bypass from this lumen into the other tissues. They may enter into other tissues, or they may, if they enter into, see, if this is a blood vessel, extravasation of tumor cells is nothing but from this blood vessel, they may come out of this blood vessel by degrading this again membrane, basement membrane, and enters into the surrounding tissue. Or else, if it forms, if the thrombus, if it gets dislodged from here and spread to distance regions, is through this circulation only. If that takes place, it will spread to the different organ which is very distant to the site of origin. So that is formation of metastasis. So once again I'll tell, by degradation of this, they'll gain access into the blood vessel. So by degradation of this blood vessel, they will enter into the other tissues or organs. And if this thrombus, if it gets moved to the other region in that circulation, through that circulation, then where does it get dislodged? From where does it get extravasated to that particular organ that will be spreaded? Okay, not only this blood vessel, they may enter gain access into lymphatic vessels, blood vessels, capillaries. To any part they can get extravasated and cause spread of these tumor cells. Okay, I hope this is clear for you. So this is all about this patterns of spread of both tumors, whether it may be invasion or metastasis. Okay.